I'm going to be recording what you see up on the screen with my voice, and if you say anything, it's being recorded too. So it records the audio and what's on the screen. And then I'm going to post it after class on YouTube. So if you ever want to go back tonight or tomorrow night or Thursday morning and refresh your memory on what we said about photosynthesis or what we said about enzymes, you can find it on YouTube. You just go to YouTube and search for Patricia Steinke and then pick our class. Or if you want to listen to me talk the same talk for another class, pick any class you want. Just make sure and pick Lab Test 2 Review. Okay? And that's just an additional optional tool that you have for studying. You don't have to do it just in case you want to do it. Okay, so I'm just going to go through the review sheet with you. And I find here on Exercise 5 Enzymes, we're supposed to identify the reactants, products, and enzyme in a chemical reaction. So I picked this chemical reaction. And the reason I picked this chemical reaction is because it's the chemical reaction we actually used for the lab that we did on enzymes. I found this chemical reaction actually on page 58, if you're wondering where I find it at. And the original reactant is called hydrogen peroxide. So you may remember the day we used hydrogen peroxide. And the day we used hydrogen peroxide, we had hydrogen peroxide break down in the presence of catalase into water and oxygen. So that hydrogen peroxide was our reactant, water and oxygen were our product, and catalase was the enzyme. And I just put a lowercase e above enzyme. How come I didn't use uppercase e? That's for energy. I reserve uppercase E for energy. So I use a lowercase e for enzyme. It's so easy to spot that catalase is an enzyme. How do you know for sure catalase is an enzyme? ACE. Nikki's right. ASE at the end. And then also we usually write enzymes on top of the arrow because they're not really a reactant and they're not really a product. So we usually put them on top of the arrow. Okay, guys, you may also notice that I have two words here, reactant and substrate. A reactant that uses an enzyme is often called a substrate. So I just wanted to refresh your memory on that. It's basically the same thing. But if it's using an enzyme, instead of calling it a reactant, we usually call it a substrate. It's the same thing. You okay on that one? Great. Okay, number 2 says define enzyme substrate active site and enzyme substrate complex. Well, I don't really care so much about that. What I really care about is if you can label pictures. So page 57, when everybody look in your book at page 57. So pretend you were given letter A on page 57 and everything was the same except all the words went away. All the words. So we would want to know how you would know which one was the enzyme. Which one was the enzyme? So how would you know that blue thing is the enzyme if the word enzyme was not there?
We learned that enzymes have a three-dimensional shape that's unique.
Okay, a unique three-dimensional shape. Anybody else want to add to what James said? Okay, it's got these depressions in it. What do you call those depressions? Yeah, so it's called an active site. Everybody find the active site there? You have to be able to label the active site. Anything else? Okay, here's what it is for me. I picked a different picture. This one I think I put on a test. Have we had this one on a test?
Yes.
Yeah. And it's from your textbook instead of your lab manual. And so I agree that the enzyme has this three-dimensional shape and it's got these receptor areas. That's the active site. But here's what else I noticed. Check out the enzyme here, here, and here. What do you notice?
It starts out together and then it doesn't change.
It doesn't change. The blue enzyme itself does not change. Okay, now check out the substrate. First of all, would you agree this is the substrate? Check out the substrate here and here. What do you notice? It does change. Substrates change into products. Enzymes don't change. So in case during the test you're thinking, I'm not sure which one's the enzyme, which one's the substrate, look at the whole picture and figure out who changes and who doesn't. The enzyme does not change. The substrate changes into product. Substrate changes into product. Okay, that's just my own personal hint to you in case you have trouble deciding. Now, once you've decided what the enzyme is, I know you can figure out the active site, right? The depressions on it. Everybody okay on what the active site is? Okay, can you figure out the enzyme substrate complex? Everybody hopes that's what I put on the test. Yes, the enzyme substrate complex. You cannot mess that up. The enzyme and the substrate are complexed. Yeah. Okay, so for number 2, first part, I really don't care about definitions. I care more about the pictures. Okay, second part of number 2. What is the effect of an enzyme on the energy of activation of a chemical reaction? Well, guys, I added this graph. You can take a look at the screen. It's an exergonic reaction. The reactants have more energy than the products, so it's going to release energy. It doesn't matter if you look at the red line or the yellow line. They're both exergonic. But one of those lines has an enzyme and one of them doesn't. The yellow one is what? With an enzyme is yellow or without the enzyme is yellow? With the enzyme is yellow. Because it lowers the energy of activation. That's perfect, Vanessa. So the answer to the question, what do enzymes do to the energy of activation? What do? They lower it. Enzymes lower the energy of activation. Enzymes lower the energy of activation. If you lower the energy of activation, you can do the reaction faster. And that's what enzymes do for us. They make the reaction go faster by lowering the energy of activation. But okay, on number 2. Number 2. Great. Okay, number 3. What happens when an enzyme is denatured? It changes shape. Yes, it stops working. Remember, as James pointed out, that enzymes have a 3-dimensional shape, and you may not remember this, but that 3-dimensional shape is held together with hydrogen bonds. Hydrogen bonds. If you change the shape, you're breaking hydrogen bonds. And so the next question is, what can cause it to denature? Well, anything that will change its hydrogen bonding. So I have 2 things listed here. What do I mean by this upward arrow temperature? What does that mean? Really high temperatures. High temperatures bad for enzymes. High temperatures are bad for enzymes. And then really low pHs like acids or really high pHs like bases, bad for enzymes. It will denature them. Denature them. And again, that means changes its 3-dimensional shape. So James was right earlier. The 3-dimensional shape of an enzyme dictates whether it can work or not work. And if it loses its 3-dimensional shape, it's not going to work. Its active site will be messed up. Okay, so that is question number 3. Question number 3. Okay, question number 4, the function of catalase. Look at page 58 with me one more time. Bottom of the page. There's catalase. Perfect. It breaks down hydrogen peroxide. It makes hydrogen peroxide break down faster. It speeds the breakdown of hydrogen peroxide. That's on page 58. Speeds the breakdown of hydrogen peroxide. Now, you would know if an enzyme is working by looking for one of its products. And in a test tube, you can't see water because the test tube's full of water. So you don't know if you're making water. So we didn't look for the product called water. What product did we look for? Because we could see it. How come we could see oxygen? It's a gas bubble. That's very good. So we looked for bubbles. If we saw bubbles, we knew catalase was working. If we didn't see bubbles, then catalase was not working. Just trying to prompt your memory on that, help you remember that. Okay, our data for question number 5. What effects do different temperatures have on catalase function? Our data is on page 60. Would you join me on page 60? Here's what I recall. We did one test tube in the refrigerator, and the temperature was around 3 or 4 degrees, right? Like today, our refrigerator looks like it's at 3.1. What was it that day? It's 2.8? It was chilly. Then our number 2, I think we had a 40-degree water bath. Is that correct? And our number 3 was 90-degree water bath. Everybody okay on those two? Okay. Look at your bubble column height. Who got number 1 as their top bubble performer? Anybody get test tube number 1? Okay. Got a few groups. Okay. Who got number 2 as your top bubble producer? Okay. Who got number 3 as your top bubble producer? Nobody. Okay, so number 1 and number 2 work pretty good. Shake your head. Number 3 didn't work at all, and Vanessa just said that's because the high temperature denatured the enzyme. So here's what I wrote. I put, cold was good, happy face. Warm was good, happy face. But hot, it wasn't good, it was bad, sad face. Why? Because Vanessa said high temperatures denature enzymes. And we've said that already once today. Must be something you have to know. High temperatures denature enzymes. So they'll work good, and then when the temperature gets too high, they're not going to work good anymore. Their function will drop off very quickly. Look on number 5. Number 5. Number 6, omit. Number 6 is omit. Everybody pays attention when a teacher says omit. I've noticed that. Say it again. What'd she say? What do I get to drop off? Number 7. What's the effect of a pH outside the optimum range of an enzyme? Will an enzyme work at its very best if you go outside its optimum pH range? No. So it's going to reduce its effectiveness. The effect of a pH outside the optimum range is bad for the enzyme. Enzyme slows down. It can't work very well. Okay, for the rest of number 7, I'd like for you to join me on page 62 and looking at our data. So we did this experiment where we had 3 test tubes. And I believe this is correct. Test tube number 1, we put acid in it. Hydrogen peroxide and catalase. Test tube number 2, we put water, hydrogen peroxide and catalase. And test tube number 3, we put sodium hydroxide, which is a base, hydrogen peroxide and catalase. Raise your hand if you had the most bubbling in test tube number 1 with the acid. Raise your hand if you had the most bubbling in test tube number 2 with water. Raise your hand if you had the most bubbling test tube number 3 with the base. Okay, so nobody chose number 1 or number 3. What did the acid in the base do to the enzyme? Denatured it. So here's what I put. You'll just take a quick look up here. I drew those 3 test tubes that you have in your notes. Test tube number 1 was catalase, hydrogen peroxide, and acid. Test tube 2 was catalase, hydrogen peroxide, and water. Test tube 3 was catalase, hydrogen peroxide, and base. And then I wrote the pHs. I hope I got this right. Was the pH of number 1, 1?
Yes.
pH of number 2, somewhere between 6 or 7? Yeah. And then pH of number 3, around a 14? Dang it. Yeah. And then no bubbling on 1, yes bubbling with a happy face on 2, and no bubbling on 3. Everybody agree? So everybody agrees that there must be an optimum pH for catalase. What was the optimum pH for catalase?
Between 6 and 7.
Between 6 and 7. Test tube number 2 was the optimum. And then again, the enzyme denatures in test tube 1 and test tube 3, just like Vanessa just said. That's her favorite word today, denature. Yes. So this drawing is just your data, just visual. Just did the same thing but visually. Okay, look through questions 1 through 7, omit number 6, and see if there's anything there you need me to repeat on enzymes. Nobody? Okay, great. Let's move on over to page 67 in our lab manual. Photosynthesis. I hope you've already boxed it. You need to know the overall chemical reaction for photosynthesis. You have to know what are the reactants and what are the products. So let's review. What are the reactants of photosynthesis? CO2 and water. And then what are the products of photosynthesis? What is CH2O? Glucose or some carbohydrate. And then what's the other product? Oxygen. So sugar and oxygen. Glucose and oxygen, or some kind of carbohydrate and oxygen. Okay, second part of number 1. Stay on page 67 to answer the question. In what plant organelle does photosynthesis occur? Chloroplast. There it is on page 67, the chloroplast. Okay, on number 1. Okay, number 2, join me on page 68. So we did this process on page 68 called paper chromatography. You may remember this was the day we had a small piece of paper we cut to fit into a test tube, and then we mashed and mashed and mashed and mashed a leaf over and over and over again. Then we poured this liquid called solvent into the test tube. And I remember we wafted the vapors of it towards our nose so we could know it wasn't water, because it just looked like water, but it wasn't water. So you may remember what it was. It's acetone. Now, I'm not going to ask you specifically acetone. What I'm going to ask you is that it was a non-polar solvent. That's what I care more about, because it was actually a mixture of acetone and ether. But to me, what's more important is that you know that that solvent was non-polar. What does that word non-polar mean?
It's
Hydrophobic. Not charged. Both are correct answers. Now, only because I have just lots of experience with plant pigments, I knew the plant pigments were also hydrophobic and not charged. So they weren't going to dissolve in water. I wanted them to dissolve, so I picked a different solvent. So it was acetone and ether, but more importantly, it was non-polar. Now the rest of the answer I found on page 68. I think we hand wrote this sentence that says paper chromatography separates pigments based on solubility. And so the question is, how does it work? It separates pigments based on solubility. In your own words, what does solubility mean?
How effective water,
Not water, here.
the absorption rate of
Is it absorbing? No. Dissolves. That's the word I'm looking for, Andrew. Very good. Dissolves. How well it dissolves. So I knew these pigments wouldn't dissolve in water, so that's why I picked a nonpolar solvent. We need them to dissolve in the solvent. And remember, the solvent was moving up the paper. So as they dissolved in the solvent, they also moved up the paper. And that's how they separated. So it's kind of cool. Okay, look on page 68, and you can see the pigments that are there. I redrew those pigments. I think I did a better job. What do you think? Kind of? Oh, well. You have to know the pigments in order. So that the pigment that went the farthest was carotene. And the pigment that got second place was xanthophyll. The pigment that came in third place was chlorophyll A. And then the pigment that's in last place is chlorophyll B. Now don't forget it's multiple choice. But you do have to memorize the order. Carotene, then xanthophyll, chlorophyll A, then chlorophyll B. Now I know verbally we went over this last question already, but I just want to go back over it with you. I have which is the most nonpolar of those four pigments? The answer is chlorophyll B. How come chlorophyll B is the most nonpolar? Like, how would you know that? Like, if you didn't know the answer, how would you figure it out? Okay, it's a nonpolar solvent. You're trying to get stuff to dissolve in it. If you dissolve the best, wouldn't you go the furthest? If you dissolve the worst, you wouldn't go very far at all. So it's related to the solubility. So if you are not very nonpolar, you're not going to dissolve very well in a nonpolar solvent. And so the most nonpolar would be carotene. Who was the least nonpolar? Chlorophyll B. So the most nonpolar was carotene, and it's super nonpolar. It goes immediately with the solvent. Wherever the solvent went to is where carotene went to. Matter of fact, I remember some of you, your carotene was on the edge of your paper. It was going off the top of your paper. It dissolved so well it was going off the top of your paper. Your chlorophyll B was had barely moved, because it's the least nonpolar. But okay on that. Great. Okay, let's go to page 72. But while we're going there, let's stop at page 70. I like that picture. On page 70, what do you like about that picture? Like, what's it trying to show you?
this invisible spectrum of white light.
White light is actually all the colors of the rainbow. It's the visible light spectrum of white light. So everybody knows white light is actually made of all the colors of the rainbow. Okay, great. On our way, let's also look at page 71. I remember doing that experiment. We had a big lamp and a big beaker of water and a flask. Okay. All right, so now we're going to go to page 72. And I remember this graph. Our x-axis is all the colors of the visible light spectrum. Our y-axis is absorption, how well the pigments can absorb those colors. And let's start with the first question. This is number 3. What color or colors of light does chlorophyll absorb the best? Blue.
Indigo, green.
I'm not going green. I'm not buying green. I'm buying blue, indigo. Some of violet's good. What else? Red is real good. But I'm not buying green. Okay, how do you know if it's absorbing the best? What do you look for? Peaks or valleys? Peaks. Look for the peaks. Okay, what color or colors of light does chlorophyll absorb the least? Green. What do you look for, peaks or valleys? Valleys. But okay on that? Okay, we had just done, when we talked about this picture, we had just done our paper chromatography, and we found that our leaves had chlorophyll A, chlorophyll B, and the carotenoids in the leaf. The leaf has all of these pigments. So that brings us to question number 4. Why is it beneficial to have several different plant pigments involved in photosynthesis? Why? Why don't you just have chlorophyll? Why don't you stop there if you were a plant? Can capture more light. That's good. I put more light means more energy is absorbed. Andrew said you could capture more colors of the white light and have more energy. So that's why plants usually have multiple pigments. Instead of just chlorophyll, they usually have the carotenoids also. And that way they're getting more of the colors of white light, which means more energy. Okay, how about the last part of number 4? Given your answer of why plants would have multiple pigments, what color of light is the best light for photosynthesis? White. Nikki's correct. Why is white light the best color?
It's carrying the most energy.
It's carrying the most energy because it's all the colors. What color of light would be the least favorable for photosynthesis? Green. How come? Do plants absorb green? No, they don't absorb green. They reflect it. So white is the best, green is the worst. Everybody okay on number 4? Number 5, true or false? Most plants carry out photosynthesis only during the day. That's true. Because plants do photosynthesis using the light. Yeah. You got to have sunlight. That's what the photo of photosynthesis is the light. You got to have some light. So the first part of number 5 is true. How about the second part of number 5? True or false? Plants carry out cellular respiration only at night. True or false? That's false. When do plants do cellular respiration? All the time. 24/7, 365. Any cell is doing some kind of cellular respiration all the time. Either aerobically or anaerobically, cells respire all the time. If they quit respiring, they die. But okay, on number 5. All right, number 6, join me on page 74. So there was this day, photosynthesis day, where we had these beakers, and in these beakers we put water and this chemical called phenol red, and the phenol red made the water kind of a pretty pink color. Then I gave you a straw. Do you remember what I told you to do with the straw? You had to blow. Remember? Bubble, bubble, bubble. And then the water went from pink to another color. What color? Yellow. Okay, what did you make in the water that made it change from pink to yellow? Carbonic acid. That's good. So I wrote that down for you. You already have it there on page 74, I believe, that water plus your CO2 from your lungs made carbonic acid. So the water started out red, and then it turned yellow. So we said that if the water is red, that meant that there was very little CO2 in the water. But if the water was yellow, that meant that there was a lot of CO2 in the water. So the color of the water told us about the CO2 level. I think we wrote that down also on page 74. Is that true? Yes. Good. Who am I missing? Elena today? Haley. You are Elena.
No.
Elena. It's over there. I got it. You're Celeste. I know that. Valerie. Yeah. Danielle. I know that. Had a moment. Okay. Put one in the window and put one in a drawer. We came back 2 days later. 2 days later. All right, look at your data for 2 days later. When we came back 2 days later, the one from the window, what color was it?
Should have gone back for red.
Was it red? Pink? It was pink. Okay, and then James said it should have gone back to red because in the window, what would the plant have been doing? Photosynthesis. And what gas does a plant need to do photosynthesis? CO2. So the plant went back, the water, went back to pink because the plant used up all the CO2 doing photosynthesis. And I even drew that real pretty for you. Look, there's my little plant. There's my pink water. I'm telling you that we had sunshine. It was so nice, yes. It's very nice, thank you. Spend time on this stuff, yes. Just want to quickly comment that this experiment did not measure oxygen. It measured CO2 because oxygen definitely changed during the experiment, but oxygen doesn't change the pH. What we were looking at was the change in pH. I agree with you, this plant made oxygen, but it wasn't the oxygen that changed the color of the water. It was the CO2. Lots of CO2 makes the water yellow, not much CO2 makes the water pink. This experiment was about CO2. Okay, then we had this other one that was in the dark. I think it was in a drawer. What color did it come out 2 days later? Still yellow. How come it was still yellow? No light, no photosynthesis. So that water was still full of CO2. It was still very high in CO2 because the plant didn't get to do photosynthesis in the drawer. Everybody okay on number 6? Okay, check out my drawing for number 7. Number 7 is, why is it important to have autotrophs, heterotrophs, and decomposers in an ecosystem? So I made a drawing. And I spent a lot of time on this too. Okay? It's a little aquarium. Check it out. I've got plants. I've got animals pooping. That's odd, isn't it? Yes. Why did she mention that? I've got rocks on the bottom. In case you can't tell, there's supposed to be little pebbles on the bottom. I've got Mr. Sunshine. All right. I've got autotrophs. Who up here are my autotrophs? The plants. And they're making oxygen and carbohydrate. Okay, now I've got heterotrophs up here. Who are my heterotrophs? My fish. Now look, my fish use the oxygen And they also eat the plant, so that means they're using the carbohydrate too. They nibble on the plants, nibble, nibble. So they're using the carbohydrate too. All right, they're using the oxygen, the fish are using the oxygen and the carbohydrate to do cellular respiration. And when you or these fish do cellular respiration, you give off carbon dioxide and water. And the plant needs carbon dioxide and water. Does everybody see I have a cycle going here? So it's clear to see why you need autotrophs and heterotrophs in an ecosystem to cycle. Everybody okay with that? Mm-hmm. In this picture, where would the decomposers be? On the rocks. That's why we put rocks in aquaria, to give surface area for the decomposers to be on. Who are the decomposers? Bacteria. That's good. Now, if the fish don't eat the leaves will get old and they'll die and they'll fall to the bottom. The fish are going to have waste products that will also fall to the bottom. What will happen to these dead leaves or the feces from these fish? They'll be decomposed by the bacteria. If the bacteria weren't there, would this be bad?
Yes.
Yes, because then the waste products would build up and eventually toxins would kill the fish and the plants. So we can see the need for the decomposer. So look at the question again. It asks you, why is it good for an ecosystem to have heterotrophs, autotrophs, and decomposers? Everybody's okay on that question? Okay, I have one more comment you may have noticed. I put a lid on my aquarium. Just thinking with me. Would the fish and the plants live longer with the lid on or the lid off? Off. Because gases from the atmosphere could come into the ecosystem and gases from the ecosystem could go back out into the atmosphere. You have to be really careful and do a lot of calculations to figure out exactly how many fish and how much plant material and how much rocks you would need to put in to be able to close this sucker up. Some people can do that. It's not me. Okay? I keep my aquariums open because I don't know exactly how much autotroph, heterotroph, and decomposers I need to completely balance it. Okay?
I think you can get monitors too, right?
You can get monitors. I know quite a long time ago, a student gave me this glass ball that was completely enclosed, and it had gravel on the bottom, it had some algae growing, it had 3 little shrimp. Yeah. Yeah, and the shrimp lasted a good 6 months. But yeah, it's really cute. Everybody's okay on that question? Super. All right, so we're going to leave photosynthesis. Do you see anything there that you need to be readdressed? Questions 1 through 7 on photosynthesis. Okay, cellular respiration. I have 3 equations I want to refresh your memory on them for cellular respiration. The first one's on page 77. Okay, on page 77, that's aerobic cellular respiration. I just rewrote it here, and I put an arrow right here because that's how I know this one's aerobic. Everybody, what am I putting my arrow on right there? Oxygen. If you see oxygen as a reactant, we're doing aerobic cellular respiration. That's aerobic. The only other thing I wanted to mention here was I hope we added how many ATP aerobic cellular respiration makes. Did you add that to your lab manual? Do you remember that? 38. I'm going to ask you that on a test. I've asked it already once. I'm going to ask it again, and it will be on the final exam. So you're going to be asked that 3 times. So you might as well get it right. If you respire glucose aerobically, you get 38 ATP. Okay, one more time. You need to be able to spot this as aerobic. And the way to spot it as aerobic is first that oxygen is a reactant, and then also that you make so many ATP, 38 ATP. Okay, and I found this on page 77. Okay, the anaerobic I found on page 78. And there should be 2 there. Join me on page 78. Now the first one is called alcohol fermentation. And it takes glucose and breaks it down into carbon dioxide, ethanol, and 2-ATP. And I circled the carbon dioxide for a reason, because carbon dioxide is a gas. So this is going to give off gas, just like aerobic cellular respiration gives off gas. And I have 2 products right here that humans use this chemical reaction for to make. We use this chemical reaction to make beer, wine, and any other kind of liquor you want to talk about. You want to talk about liquor? We're talking about this chemical reaction. You get different kinds of liquors, beers, or wines depending upon what you ferment. You know, what your glucose source is. Anyway, which product, carbon dioxide, ethanol, or ATP, are humans trying to use this chemical reaction to make if they're making beer, wine, or any other kind of liquor? What are you going for? What product do the humans want? Alcohol. So for making beer, wine, or any other kind of liquor, that's what we're harvesting right there. The alcohol. It's called ethanol. It's a 2-carbon alcohol. You guys heard of it? Ethanol? Yeah. What if you were trying to make bread? Are you trying to get the CO2, the alcohol, or the ATP? If you're a baker, which product of this chemical reaction are you after?
Oh, baker,
CO2.
CO2.
Because you're trying to get the bread to rise. James is right. The bakers are interested in the CO2 production. The distillers, the brewmasters, they're interested in the ethanol production. Now, to make this happen, you're going to have some cells, because this is cellular respiration. What cells are the bakers and the brewers going to put in with the glucose to make this happen? Yeast. It's yeast that does this. How do you know, I'm just talking about this one right here, how do you know this chemical reaction is anaerobic?
The amount of ATP.
The amount of ATP being made is only 2, and I heard somebody else tell me, how do you know? No oxygen. Oxygen is not a reactant. It's anaerobic. But okay on that one. Okay, we added in the next one. Maybe right underneath the sentence, when animals such as humans ferment, they produce lactate instead of ethanol and carbon dioxide. Did y'all add a reaction there? Yes, Celeste said we did. Anybody else have it? You guys have it? I might have called this lactic acid or lactate. It's the same thing. You know when you are working out and your muscles start burning that you have gone anaerobic, that you're making an acid and it burns in your muscles. And then you're probably like, oh my God, why'd you write sour cream underneath my muscles burning? Because this is a product you can make with this chemical reaction. We use bacteria that can do lactic acid fermentation. We add that bacteria into milk, cream, and it ferments and it makes sour cream. It ferments and it makes yogurt. What else do we make? Soy sauce. You can add it to soybeans and let the bacteria ferment the soybeans, and you get soy sauce. You can add it to cocoa beans and let the bacteria ferment. You get chocolate. You can add it to vinegar and cucumbers. Add the bacteria to vinegar and cucumbers, and what do you get? Pickles. You can add it to olives and salt water, and that's how we get the flavors of the different kinds of olives that we like to eat. You start thinking about it this time of day, it's just yummy, just yummy. Blue cheese. Oh my gosh, blue cheese. Ready? Okay, on just these two reactions being anaerobic, they only make 2 ATP. Okay, super. Let me go back to this question. Number 2. It doesn't matter if I look on page 77 or 78, the answer's the same. Both anaerobic and aerobic cellular respiration ordinarily begin with what molecule? Glucose. Y'all check it out. Glucose is a reactant regardless of the chemical reaction you look at. It's the reactant in aerobic cellular respiration. It's the reactant for anaerobic cellular respiration. So the answer to number 2 is glucose. Glucose. Number 3, in which reaction is glucose completely broken down? Well, I'm going to give you a hint. If glucose is completely broken down, you get the most energy out of the glucose. So which one? The one where you get 38. So which reaction is that? Aerobic cellular respiration. Aerobic cellular respiration. In which is glucose completely broken down? Aerobic cellular respiration. And how you can tell is because you get the most energy out of the glucose. You've chopped it up and harvested all the bond energy out of it. Okay, everybody knows the answer to the second part of number 3. How much ATP does each reaction yield? 38 for aerobic, 2 for anaerobic. I promised to ask you that question. But okay, on question 3. Okay, question 4 is the fermentation experiment. Well, first of all, I drew it up here. And in case my drawings don't look great, I set some up here too. This is where we had itty-bitty test tubes, and we put sugar and yeast in the itty-bitty test tubes. And then we had to shove those itty-bitty test tubes into a bigger test tube and do the quickie flip thing. Y'all remember doing that? Then they went to the incubator, which was nice and warm. We came back about 20 minutes later, and we measured the gas production in each one. Okay, our data is on page 82. Tube number 1 was glucose and yeast. Tube number 2 was fructose and yeast. Tube number 3 was sucrose and yeast. Tube number 4 was our negative control, no sugar. Instead, we used water and yeast. Yeah. Now, I might be wrong, but did glucose and fructose, was fructose first place, glucose second place, and sucrose third place? That's how it was? Okay. Now, glucose and fructose usually do pretty well. Sucrose, I think, did okay that day also. But glucose and fructose usually do better than sucrose because chemically, glucose and fructose are simple sugars. Now, I didn't write that down up here. What did I write down? Mono. What does that mean? One. Simple sugar. And so the cells, the yeast cells you put in there could quickly just break down the sugar and get the ATP out of it. So it was real fast. Now, obviously, from your data, sucrose could be used also. But what does a yeast cell have to do with sucrose before it can use it for energy? It's a disaccharide. What would it have to do? Break it apart. It's 2 sugars linked together, so it'd have to break the 2 sugars apart, then use the 2 sugars that it broke apart. So sucrose works great, but it takes a little longer to ferment it. Okay, here's what I will ask you. We measured gas. There's a gas bubble right there, and the question is, what is that gas? CO2. I'm going to ask you what was the gas at the top of the tubes, and it was CO2. Because these cells are doing cellular respiration, producing CO2. You're going to have to know the name of the gas is CO2. That's the last part of number 4. What was the gas? CO2. But okay, on number 4, the cells fermented the sugar. They made CO2. They fermented the sugar. They broke down the sugar, if you like that better. They broke down the sugar. The cells were trying to get ATP out of that sugar. Per glucose, how many ATP can a yeast cell get out of a glucose? 2, because we're doing fermentation. Is fermentation aerobic or anaerobic? I'm listening. Fermentation aerobic or anaerobic? Anaerobic. So they can get 2. 2's better than nothing. Okay. The aerobic cellular respiration experiment, I didn't try and draw, and you can see why. You can't draw it either. Okay. Yeah, it would be a challenge. There were these big tubes. We put stuff in the big tubes, and I didn't wrap this tube completely with aluminum foil so you could see inside of it. But the day we did it, we did wrap the tubes completely with aluminum foil, because what we put inside the tubes were plants, and we didn't want the plants doing photosynthesis. We only wanted them doing cellular respiration. If you would, join me on page 79 to see the 3 different tubes that you constructed. Guys, this was the day with the shaving cream. In case you're having a hard time remembering, shaving cream sticks in your minds. Yeah, shaving cream. So in the first test tube, we put germinating peas. In the second one, we put dry peas with glass beads. And in the last one, we put just glass beads. So obviously our last one was our negative control. So we have a couple questions to answer on the aerobic cellular respiration experiment. Which is question number 6. Which direction did you expect the shaving cream to move during the aerobic cellular respiration experiment? Did you expect it to move in or move out? In. And the reason why is that we put these respiring cells inside of the test tube, and those respiring cells should have been using up gas, thus creating a vacuum. If you don't like the word vacuum, you could use the word suction, because they were using up a gas, and that would have created a suction. Thus, the shaving cream would have moved inward. What gas were the respiring cells using up? Make up your mind. I heard CO2 and oxygen. We're doing cellular respiration. What gas do you use up, CO2 or oxygen? Use it up. You don't give it off, you use it up. Oxygen, definitely. It's oxygen. We're using up oxygen. Okay, just pretend I put you in a sealed off room. Eventually, Kevin would die. So would you, because what gas would you use up? Oxygen. But okay with that? Super. Would you quickly glance back at exercise 7, questions 1 through 7, and see if there's anything there that you needed repeated? Everybody's okay? Number 5? I was just showing it to you. Yeah, just kind of get a new visual lock on it. So like if you see this on the test, you'll remember it. I just wanted you to see it and remember it, okay? It's been a few weeks ago. Anybody else? Okay, great. On mitosis, guys, I'm not going to go over interphase, prophase, metaphase, anaphase, and telophase because you just did this little handy-dandy extra credit sheet that pretty much goes over that. And I just want to tell you that if you'll go back and review that, that would be very useful for mitosis. But I do want to talk to you just a little bit about the cell cycle and a few vocabulary words just to make sure you've got it. The cell cycle is on page 86. Page 86. So if you will join me on page 86. So there's this purple area at the top of page 86 called interphase. Interphase. And that is subdivided into G1S and G2. And then the rest of the cell cycle is basically mitosis, prophase, metaphase, anaphase, telophase, and with the associated cytokinesis. But okay, so far on the cell cycle, what I'm talking about. Okay, great. So everybody glance up at what I did up here. I took your cell cycle and I drew on it. Let me show you what I drew. First, I drew in red. But I drew chromatin and chromosomes. And I remember drawing this for you on the board one day. The difference between chromatin and chromosomes. Well, guys, it's DNA, but one of them is long and spread out, and one of them is tightly coiled. It's the same thing. The one's spread out and one's tightly coiled. Which one's spread out, the chromatin or the chromosomes? Chromatin. Chromatin. So I wrote that, and this is chromatin. And then look at the top of the page at the cell cycle. Do you find chromatin during interphase or during mitosis? Be all spread out during interphase, or would it be tightly coiled during interphase? Which one? Interphase. It's chromatin. So if you glance up at my drawing, I'm sorry, I kind of wrote it down for you. But this is chromatin, and that happens. We find it during interphase. Now, what is this? G1S, G2. Why did I write that? Because that is interphase. Correct? Okay. Now, this is a chromosome. You wouldn't find chromosomes during interphase. When would you find them? In mitosis. I didn't write mitosis. Look what I wrote. What is that?
That's peanut.
Prophase, metaphase, anaphase, telophase. It's mitosis. And those are just the parts of mitosis. Just like G1, S, and G2 are the parts of interphase. Correct? Is okay on that? All right, now I can see a little confusion because what I have up there is a little different than what I drew here. What's the difference? Both of my drawings. Something's different. This is single-sided chromosomes. These are double-sided. Yes? Okay. Find it in the cell cycle. You tell me. When would this single-sided piece of chromatin become double-sided? During S. Okay? Okay, now I want you to think hard. I'm going to go through each one of these and you tell me, would a chromosome be double-sided or single-sided? We'll start with prophase. You were in prophase. Would a chromosome be double-sided or single-sided? Double. How about metaphase? Double. How about anaphase? Single. How about telophase? Single. So I want to back up and I'm going to show you that I did that. During G1, I'm single-sided. During S, now I'm double-sided. I stay double-sided. I stay double-sided. I stay double-sided till there. In anaphase, now I'm single-sided, 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 double. So I starred S for a reason. And I starred anaphase for a reason. Why did your teacher star those? That's where there's the change. I go from single to double at S. I go from double back to single at anaphase. And it just keeps going and going. It's a cycle over and over again. But okay, on what I drew up here. Okay, so speaking of chromosomes, you have to know the parts of a chromosome. So stay on that same page, page 86, and look at the bottom. The bottom.
Checkpoints beyond the test.
Checkpoints are not going to be on this test. Checkpoints are going to be on the lecture test. Good question, Adrian. Okay. Did it again there in blue. Okay. First of all, I drew a simple drawing of a chromosome. Your book is way better than mine. But I did label chromatids and centromere, and those are the two parts I need you to know. In your own words, how would you describe a chromatid? Isn't it one side of a double-sided chromosome? One side of a double-sided chromosome? Okay. In your own words, how would you describe centromere?
Center.
The center where the two chromatids are joined. It's where the two chromatids are joined. Centromere. You okay with that? All right, now, you can glance, and I'll tell you where I'm at in our review real quickly. I'm on number 2. And right now I have chromatids and centromere complete. And I also have the last part, identify the parts of a chromosome. We're going to do these other words in just a minute. But before I leave the screen, I don't want to miss this opportunity to talk about another term that isn't until the back page. It's not on the front of this page, it's on the back. It's words you know. Why do I have a red one and a blue one? What am I trying to show? Maternal and paternal. So what do you call a pair of chromosomes that have information for the same traits? Homologous. I'm trying to show you a homologous pair of chromosomes. They're about the same size and about the same shape, but one came from my mother, one came from my father. We call them homologous. I'm showing you a homologous pair right there. Homologous. Okay, I went ahead and did the other 3 words. I know you guys know this. Cytokinesis is division of the cytoplasm. Now, let me ask you this, because a few people missed this on this mitosis assessment you did. What stage of mitosis usually do we see cytokinesis occurring in? Telophase. A few people missed that. Telophase is where you usually see cytokinesis occurring. Okay, spindle fibers are the small proteins in the cell that pull the chromosomes apart. They do their job in anaphase, when we pull the chromosomes apart in anaphase. And then lastly, organelles that we find only in animals. We're not even sure what they do, but for some reason we talk about them when we talk about mitosis. They're called centrioles. There's always 2 of them. We're not really sure what they do, but we do know that they copy themselves, and then the pairs separate away from each other so that each new animal cell gets a pair of centrioles. So centrioles are only found in animals. So just glance with me on number 2 and make sure that you're okay with all the vocabulary that's listed on number 2. Everybody's okay on number 2? Okay, number 3. Join me on page 88 and 89, and I'm looking for 2 things that are different between plant and animal mitosis. And I'm going to go ahead and grab the easy thing. And you have to come up with one other difference. The easy thing that we just mentioned, it's still up on the screen, is that animals have centrioles and plants don't. I'll ask you that. Animals have centrioles, plants don't. So animals have to make sure and divide their centrioles into equal cells whenever they're doing mitosis. Plants don't have to worry about that because they don't have centrioles. Don't have to worry about that. Okay, that was one difference. I need another difference between plant and animal mitosis. Cleavage furrow and cell plate. You guys are on it. Cleavage furrow and cell plate. The way they do cytokinesis is different. So I just made a quick little table here. Plants have no centrioles and they use a cell plate. Animals have centrioles and they use a cleavage furrow. I can't even believe I'm asking number 3, second part. Why can't plants use cleavage furrows? Like, why can't they? Cell wall. And cell walls are rigid. Yeah, cell walls are rigid. Oh, look, I did drawings for you. Would A be, look at my choice right here. Would A be plant or animal? Plant. B? Okay, and then what is this? Cell who?
Cell wall.
Plate. And what is this? Cleavage furrow. It's okay on my very simple drawings. You guys know you like my art. I can't believe you act like you don't. Yeah. Yeah, I could like market this stuff. It's so good. I know. I could retire early. Biology teacher makes it big as artist. All right, number 4. So on the test, I'm going to test you on mitosis in 3 ways. Let me outline those 3 ways. Slides. And so the slides are going to look similar to page 88. You guys remember one day we did slides where you had to find the stages of mitosis in animals. I walked around and you showed me. Then you had to find the stages of mitosis in plants. And you walked around, or I walked around and you showed me. And then again, just reiterating that we had a few pictures here on this handout that were pretty useful. Most of you were good on figuring out the stages of mitosis from this handout also. Okay, so that's what I mean by slides. Okay, then also we spent quite a bit of time building pop bead chromosomes and simulating the stages of mitosis on our tabletops. Some of you may have taken pictures of that with your cell phone to go back and study. So that's way number 2. I'm going to test you on mitosis. Lastly, models. I've got these great models back here behind Elena and Philip. The blue models clearly are animal mitosis. You can tell by the cleavage furrow. And then these really great models right behind Philip, those are plant cells. Those are deluxe models. They're just impressive. It's like you'd be liking those, yes. Make sure and stop by and look at the models for the test. But okay, on mitosis. All right, lastly, meiosis. I'm on the back. Okay, let's play multiple choice on these 3 vocabulary words that are on number 1. This is a cell with 1 set of chromosomes. Haploid. This is a cell with 2 sets of chromosomes. Diploid. These are pairs of chromosomes with the same traits. Homologous. Yes. What do you use for diploid? A shorthand way to show diploid. Shorthand. 2N. And I think I've said it enough times, so I'm going to go ahead and ask you, what's the diploid number for humans? 46. Did you know diploid number for humans, 46. 2N is diploid. Diploid cells have homologous chromosomes because every maternal chromosome has a paternal chromosome that's matched with. So diploid cells have homologous chromosome pairs. They have 2 sets, a maternal set and a paternal set, and every individual maternal chromosome has a matching partner that's paternal. Except if you're a little boy. If you're a boy in the classroom, you have 1 pair of chromosomes that don't match up. Thanks for coming. What about talking about boys have 1 pair of chromosomes that do not match up? Yeah, they have X and Y. So men are not completely homologous. Women, you are completely homologous. Completely matched up. Food for thought. But okay, on the 3 definitions there. Okay, then the stages of meiosis are on page 96 and 97. Meiosis is 2 divisions. Each division has a pro, meta, ana, tele. Okay. Okay, join me now on page 101. Man, I like that page. Page 101. The top is making sperm. The bottom is making eggs. We talked about this downstairs today. Per original diploid cell in the testes, how many sperm can you make? 4. Per original diploid cell in the ovaries, how many eggs can you make? 1. Say it again.
The rest are polar bodies.
The other 3 are polar bodies. What happens to polar bodies? They die. That's good. So everybody's good on that. Okay. Can you recognize an ovary from a testes? I showed you downstairs today again. Make sure you can recognize an ovary from a testes. And just think about how I can mess you up on this, okay? If you don't know an ovary from a testes, first you'll miss the first question, name this organ. Then the next question will be, per original diploid cell of this organ that starts meiosis, how many functional sex cells can you make? If you think it's an ovary and it's a testes, you're going to miss the ovary-testes question. There's the first one you missed. Then you're going to pick 1 egg when the answer was 4 sperm. So you're going to have to know the ovary from the testes. Make sure you can tell an ovary from a testes. Okay, on the ovary slide, everybody look on page 102. There are these big hollow things full of fluid called follicles. And the egg is inside of there. If you think about it, they kind of look like there's an egg in there, if you think about it, huh? Remember, those follicles are going to swell and eventually they're going to pop and the egg will be released. Whereas in the testes, thank goodness those don't swell in first. That would be amazing. The testes have these tubes inside of them called seminiferous tubules. And everybody listen to that word. I'm trying to make it where you can remember it. Seminiferous. Does everybody hear semen? In seminiferous. Semen. And you guys know semen are male reproductive fluids. Seminiferous tubules. Those are those tubes that look like donuts because we cut them in cross-section. They look like donuts. They're making sperm. Okay, now check out the picture, page 103. On the bottom right-hand corner, right above where it says 100 micrometers or 100 micrometers, do you see this little triangular patch of blue cells? Those are interstitial cells. They don't make sperm. What do they make? Testosterone. Yeah, so that's what your testes look like in cross-section, men. Ladies, that's what your ovaries look like in cross-section. So you got to be able to tell an ovary from a testy. Okay, talked about that enough. About that. Number 5, know the stages of meiosis. Okay, I do care about this. Synapsis. Okay, so would you please, in case you don't remember this, would you please go back to page 98 and look at prophase 1. Synapsis. What is synapsis? Look how they wrap around each other. The homologous partners find each other and then they wrap their arms around each other. So just look at the picture. You can see they're synapsing there in prophase 1. And then while they're all wrapped around each other, do you remember enzymes cut them and then enzymes reseal them so that what could occur? Crossing over. So what is crossing over? Switching of pieces of DNA between the homologous partners. How can you visually tell if they've done that thing called crossing over? The colors, yeah. So like the blue chromosome will have a little red, and the red chromosome will have a little blue. So you see a switching of the colors. And we know that we've done crossing over. Okay, now scientists have learned, anything that's doing sexual reproduction is doing crossing over. So then they thought, why? Why would you do crossing over? What does it get you? Variation. Okay, now we should have done all of exercise 9 downstairs. Class, I do want to highlight number 1, allele, genotype, phenotype, heterozygous, homozygous. Those vocabulary words are on this test. I know we just briefly touched upon them. We'll keep talking about them in lecture. Lucky for you, your lab test is after that, so you'll get to practice some more with all those vocabulary words before our test. But make sure you know those vocabulary words. Make sure you know how to do chi-square. Anybody need anything repeated? Okay, so what are you going to have to bring?